Okay, well, thank you for the invitation, and thank you all for coming. Today we're going to talk about food and some of its manifestations. But I want to take a sort of historic perspective to begin with and start with reminding you that once upon a time we got our food by either hunting or by gathering, by going out and collecting what nature has to offer. As it turns out, we still do that. If you go to a main source of our food, which is a supermarket, you will be greeted, of course, by long aisles of what are basically very, modified versions of wheat or rice or corn. And how do we acquire this food? We gather it. Except we gather it using money, because only a very small fraction, at least in the United States, only a very small fraction of our population actually produces food. Right? We take advantage of their efforts, but we gather it with money. And I like to say that our entire economic system of not just the United States, but of any country, is really revolves around the acquisition of food. That's what produced the economic system we had. If you don't believe me, just wait until the cost of food escalates to the point where you can no longer afford it or where it doesn't exist. All of a sudden you have revolutions, the government's overthrown, economic systems change. So if you doubt that food is at the fund basis of our economic system, just see what happens when you don't have it. So I thought I would say, since we gather food by using money, let's see where we gather it from. So this is where we gather it from. This is the market value of agricultural products sold in 2007. And I show this for one reason. Of course, it's no surprise. This is one dot equals $20 million worth of agricultural value. I show this, of course, to emphasize things you know already, which is the Central Valley is a major agricultural place, and of course, the Midwest. But look at this. There's Philadelphia. Look at that. What's that? That's Lancaster County, right? Lancaster County, and unless you think that agriculture is diminishing in importance in the United States, here are some recent data from Lancaster. This is the difference between 2007 and 2012. There's been a 4% increase in the number of farms, 3% increase in the land in farms, 38% increase in the value of market products sold, and interestingly, in Lancaster, and this may be maybe not unique to Lancaster, but it may be because of a large Amish population, relatively a small amount of money is obtained from the government. In Midwest, a lot of the income of farmers actually comes from government subsidies, but not in Lancaster. Here's where we rank, just for your information. It's no surprise, perhaps, that Lancaster County amongst counties in the state of Pennsylvania is number one in almost everything, certainly in terms of the total value of agricultural products sold, as I just showed you. Here's where it ranks in the United States, where Lancaster is number 15th in terms of total value, number one in terms of layers. So basically, a lot of eggs. So if you're buying eggs in Philadelphia, they probably came from Lancaster. Okay, so just to give you some context, right? So 1 hour west of here is this enormously productive, valuable agricultural community. So it's really quite remarkable. So this is an outline of what I'm going to talk about today. I'm going to talk briefly about how humans interact with food. We have a very complicated and, as I say, completely and totally irrational interaction. We make our food decisions based on no logic whatsoever. I'm going to tell you that almost everything we eat comes from someplace else originally. And I'm going to tell you that what we basically eat are mutant derivatives of an ancestral types of plants and animals. And I'm going to tell you where those mutations came from and what we're doing nowadays to genetically modify our food. Okay, so let's talk about our interaction with food. Okay, and when you go to the supermarket, you're greeted, of course, as I showed you just here, with this aisle and aisle, miles and miles of food. And to illustrate just how we interact with food, consider these breakfast cereals and what is on the box to try to get you to buy it. That tells you what we consider important. Well, this one says it's USDA organic. This one says its filling is made with real fruit, implying that there's something called artificial fruit, I guess. It tells you that this is a non-GMO product verified, so people obviously must care something about whether their food is modified in a laboratory. That it's got 46 grams of whole grains and 6 grams of fiber. Basically, somehow these things presumably appeal to some fraction of our population. This is my favorite example. When we drive, we have a second home out west of Lancaster, and we like to stop and get Roy Rogers chicken on our way there. And as we were standing in line waiting for our chicken, I noticed that they had these chocolate chip cookies for sale, Lucy's. I was struck by the fact these were gluten-free and vegan, all natural, no peanuts, tree nuts, milk, eggs, non-GMO, 0-gram trans fats, certified. You would think there was nothing in this bag until you turn it over and look at the other side and see the list of ingredients. Here's the opposite end of the spectrum. That's sort of processed food. Here's all natural food sold by Shady Acres Farm, which we like to frequent, run by Mennonites, and it's an organic farm. And they sell these eggs. One does say these are unclassified brown eggs, meaning to say they're not classified according to size. They're GMO-free, which means there's no such thing as a GM chicken, but these are not even fed on GM corn. So they're basically fed on organic, non-GM corn. They're free-range, grass-fed hens. Okay, so they're running around. They call them grass-fed, which I think is interesting because chickens don't actually eat grass. They eat the insects and stuff. The implication is they're running around in the grass. And it tells you when the eggs were laid. What more could you want to know about your eggs? This is my favorite cartoon from the New Yorker. Did Jesus create these locally? I think that about sums up our attitudes about food, right? For many people, food is a religion. It really is a religion. If it's not a religion, it's your source of identity. You ask people, what do they say? I'm a vegan. Just like somebody would say, well, I'm Asian, or I'm Vietnamese, or I'm British, or I'm whatever. I'm a vegan, I'm a vegetarian. No one says I'm an omnivore, or no one says I'm a carnivore. But we have other sources of identity. So where did I take this picture? Anybody have any ideas? New York City? Reasonable guess? France. Sometimes students say Philadelphia, Italian market. I took it in Hangzhou, China. Right? Hangzhou in China, near Shanghai. The take-home message is I could have taken this picture anywhere in the world and it would look the same. Right? Because we eat the same thing everywhere in the world. Pretty much. Of course, there are differences, but pretty much what we eat is very similar. Now, what we eat actually has a home. It originated in particular places in the world, and these are the places where some of the food we eat originated. Of course, probably most everybody's familiar with the fact that wheat and barley arose in the Fertile Crescent, as did rye and grapes. You may or may not know that things like potatoes originated in peanuts, and beans originated here in the Andes, and then, of course, corn, and tomatoes, and dry beans originated in Mesoamerica. Not surprisingly, rice originated in Southeast Asia. They don't have chickens originated here. Oranges, believe it or not, some people have a hard time believing because we think of oranges as a temperate crop, actually originated in Southeast Asia. Sugarcane here. So the only thing that originated in North America, strawberries are actually a hybrid between two plants grown here and here. Sunflowers are basically, and blueberries, are native to North America. This is why those crops originated in those parts of the world. They originated there because that's where agriculture originated, right? So these crops were domesticated and became part of our diet in places in the world where people started to cultivate plants intensively, producing what we now call agriculture. Okay. It used to be thought back when I was in college, I'm sure back when you were taking, maybe if you ever took an anthropology course in college, you probably learned about the diffusionist idea of, that agriculture was such a complicated thing, it could have only been invented once by Westerners and diffused from the Fertile Crescent around the world. That's what anthropologists used to think. But of course, that isn't true. It actually was invented, or however you want to call it, invented, discovered, created, independently in many different places in the world, and this is probably just a subset of the places in which people learned how to grow crops. In fact, if you read the literature now, agriculture is a horrible way to live. Anybody who's ever been a farmer knows this. I used to work on corn, and I can tell you, it's not fun being a farmer. It is definitely not fun. There are weeds, it's hard work. It was much easier back in the day when all you had to do was go out and gather your food or hunt or whatever. People have done studies and they figured out how long it takes to acquire, how long does it take the tung to people in Africa to collect food, and it turns out it's not that many hours per week. Okay, so the invention of agriculture, and you only have to read, Genesis to hear about, learn about this. I mean, basically, being a farmer is not a good lifestyle. But the creation of all these domesticated plants around the world and the recognition that plants came from not just locally from Europe, but were found in other parts of the world, of course, gave rise to the age of discovery, when Europeans, of course, were interested in going and collecting spices, pepper or wherever, or mace or whatever from Southeast Asia, or the great voyages of this period were really largely initiated or by the desire to find new plants, mostly plants, possible, to enrich your life, basically. So it had a huge impact. And one illustration of just how complicated the history of the food we eat is this picture of how the history of coffee. So coffee originated in Africa, specifically in Ethiopia. Arabica coffee is actually a allotetraploid. It's a cross between two different species of coffee, so it's a tetraploid. And it originated in Ethiopia, and then, of course, it got the name Arabica coffee because the coffee culture really sort of developed in Yemen and Arabia, and then seeds were taken to the Netherlands by the Dutch, and of course the Dutch, were explorers, and they took it around the world. In one place they gave some seeds to the King of France, King Louis XIV, and he distributed to various places, including Martinique, and so on and so forth. So this is just an example of how one crop has moved around the world as a result of the explorations, if you will, the activities of, in this case, the Europeans. So what is it that we eat? Well, we eat two major groups of plants, the cereals and the legumes. The cereals are these plants right here. These are the major ones, corn or maize, wheat, rice, and barley. These are all the same family of plants, very closely related. They, the same thing as growing in your front lawn. They're grasses, same family of plants. Okay? And so this is the main source of our calories. We either eat these directly, we get our calories directly by eating them, or indirectly because this is what our animals eat. Right? Most of the corn the United States produces is fed to livestock. Okay? And here, of course, are the other major plant that we consume are the legumes. Okay? Chickpeas and peas and lentils and beans and such things. What do we eat in the form of animals? So the major animals we eat are these: cows, chickens, pigs, and sheep. Understand that these were selected from a much larger group of large animals that could have been used for agriculture, but these animals have the psychology, basically, the behavior that allows us to domesticate them. Because when it comes to domesticating animals, it's all about behavior, whether or not an animal can live in association happily with a human. If animals can't live in association with humans, end of story, no domestication. So I like to show this, what's wrong with this picture, just to show you what we've done now. So there are several things that are wrong with it. Firstly, if Adam and Eve ever existed as, they were definitely not white, right? They were black, because humans came from Africa. So this much we know. Whether you agree with the biblical story or not, we know that the first humans were black. So that's but the other thing that's interesting about this picture, which was painted in the 19th century in the Vatican's collection, is that a lot of the animals that are shown here, at least the form in which they are shown, did not exist at the time life arose. Right? They're relatively recent creations. They're domesticated. They've been modified by humans over the last 10,000 to 15,000 years or so. All right? So these, in fact, were not created by the hand of God. They were definitely created by the hand of humans. Okay. If you want evidence of this, we can also look here in 1645, another well-known painting, and I draw your attention to this watermelon. I bet you've never seen a watermelon that looks like that, right? Where the locules have got spaces in them. We're used to watermelons that are completely filled with flesh. Okay, that's something that we have done to the watermelon over the hundreds of years that we've been domesticating it. We've actually changed even more. I mean, the ancestral watermelon is a little tiny fruit about this big, hard as a rock, basically. So we've modified it dramatically. And since this time, of course, we've done interesting things to watermelons. We've, certainly increased the amount of flesh that they have. We've changed their colors, and we've made them seedless. To the geneticists in this audience, which I'm sure there are some of you, how do you get a seedless watermelon? You can ask that question. If you don't know the answer, we'll answer that in the discussion. I always ask my students that question. Where does the seedless watermelon come from? Think about it. Okay. How do you propagate it? That's the question. Okay. So domestication of plants and animals was very important for Darwin's thinking. There's a chapter in the Origin of the Species devoted to this. He wrote a two-volume two volumes on animals and plants under domestication. Darwin was well aware, because he was a pigeon breeder, well aware that humans can dramatically change the appearance of the organisms they interact with. Okay? And of course, that was part of the evidence for the idea that evidence for natural selection. One of the examples that everybody's familiar with, I'm sure, of course, is the dog. The dog evolved from the wolf, and humans have modified dogs for a variety of reasons, initially for work purposes, but now we can impose our aesthetic on these animals as we have. I like to say that man is dog's worst friend. If you own a bulldog, you're nasty. Those poor animals are suffering. Suffering. So we have we live near Schuylkill River Park, and here's our local dog walker, and he's got, just to show you the diversity in dog breeds, you can get whatever you want at Alice's restaurant, and if you want a dog breed that somehow meets your aesthetic. So let's look at an example of this. So this is similar to the ancestor of modern sheep. Goats actually look pretty much the same way. Okay, like any wild mammal, if you ever have mice in your house, you know this. The wild mammals have an agouti, a brown coat color, short fur, right? Okay? But not our domesticated animals, of course, because when we think of sheep, that's what we think of, right? We think of animals with white or black wool, or hairless or woolless, or long curly wool or whatever, okay? Various modifications that humans have created over the years of domesticating and breeding these animals. And we know what the genetic basis for some of these things is. For example, white wool coat color in sheep, as it is in virtually any animal, including the yellow mouse, is a mutation of the agouti gene, overexpressed. So mutations that cause this regulator of melanocyte differentiation to be overexpressed, okay, cause, I won't go into the details, but cause basically a sort of a whitish or yellowish coat color. Okay? This mutation probably occurred, 10,000 years ago, whenever sheep were domesticated, right? So it occurred spontaneously and was selected. And why did humans select, white wool or long wool? That's an interesting question, right? Why was that mutation interesting to humans? Who knows? But it was. Our milk mostly comes from this breed of cow, the Holstein. It's the most productive breed in terms of milk production of cow in the world. An average Holstein produces about 9 gallons of milk per day. The all-time leader is this girl right here, Burwell Buckeye Gigi. She produced 29 gallons of milk per day. Now, how much milk do you think a cow needs to produce? Right? A cow needs to produce just enough milk for her calf. 1.5 gallons. Right? So we have engineered these animals way beyond what Mother Nature intended. In fact, you don't milk these animals, they are very unhappy. Forget about it. They're gone. Right? So these animals are our creation. Right? They're way beyond what Mother Nature intended. Here's the plant example that we like to use. Anybody recognize this plant? Right. Brassica, all of these are the same species, Brassica oleracea, right? Cauliflower, these are 2 mutations that affect the differentiation of flowers, the so-called cauliflower genes. Broccoli, kohlrabi, these are mutants in flower differentiation. This is a mutation in the stem. Of course, it doesn't elongate, it gets larger. These are leaves here. Of course, the kales and cabbages are mutations in various sorts of leaf shape. Anybody know what part of a plant a Brussels sprout is? It's an axillary bud, right? If you've ever seen a Brussels sprout, ever grown a Brussels sprout plant, you know there used to be a leaf right there. And this is a little bud in the axil of the leaf. It's a branch, okay, that just didn't elongate, right? And here's a cabbage where the stem doesn't elongate. Here's another example of human genetic engineering. That is teosinte. That's what we recognize as teosinte, right? So the mutation, and going from here to here took about 5 mutations. There are basically 5, I mean, of course there's lots of modifiers, but there's 5 major genes that were 5 major mutations or alleles that were selected during the domestication of maize. John Dobley at the University of Wisconsin worked this out. It's an amazing story. Okay. Actually, the number 5, George Beadle, I love this. I teach genetic analysis at graduate level class. And, you know, George Beadle, Nobel Prize, one gene, one enzyme, very famous president of the University of Chicago and Caltech. He was a maize geneticist in his first life, and when he retired, he went back to being a maize geneticist after doing all these wonderful things. And he decided to figure out how many genes were involved in the domestication of corn. So he did a very simple genetic experiment. He crossed teosinte by corn. Corn evolved so recently from teosinte, they're still cross-compatible. So he crossed, teosinte, he crossed this by this, and he made an F2 population. Right, just let it self. And then he counted in that large population how many plants looked like maize and how many plants looked like teosinte. And based on the frequency of those extremes, he calculated it had to be about 5 genes. And he was right. John Dobley then came back later and actually mapped those genes, did exactly the same experiment with the modification that he mapped them with molecular markers and showed, lo and behold, 5 major loci. And then he went ahead and cloned them. Okay, the changes that occur when plants are domesticated are so common, they happen all the time, that it's called what's called the domestication syndrome. So these features are characteristic of all domesticated plants. One of the very first things that happens is you have what's called loss of seed shattering. Wild grasses and wild plants in general do not retain their seeds, right? They are adapted to spread their seeds into the wild, right? But when humans start cultivating, start harvesting crops, they unintentionally select for mutations where the seeds don't fall off the head, right? Because any mutation that kept the seeds on the head, that blocked the shattering trait, would naturally propagate more quickly because you would harvest more seeds from that plant. So you can imagine this occurring unconsciously. Loss of seed dormancy. We take for granted that when you buy a packet of seeds from your local supermarket or whatever and you plant them, you expect those things to germinate in about a week or 10 days. But of course, wild seeds may or may not germinate. I like to plant black walnuts to increase my black walnut population. I plant about 5 or 10 black walnuts in one spot, and maybe I'll get 1 seed to germinate. Or 2, and maybe they'll germinate over a couple of years. Compact inflorescences, large seeds and fruits, photoperiodic responsiveness, uniform flower, and loss of toxic chemicals. All of these things occur when we domesticate plants. So what's the source of the genetic variation that we have unconsciously or consciously selected for as we've domesticated and modified our plants? It has 3 different sources: naturally occurring spontaneous mutations. And this can either be gene mutations with single large effects or polygenic effects, induced mutations, so we can create mutations using X-rays or chemicals, and of course nowadays we can add genes to plants. We can use and I should put one more, put CRISPR down here, which is a sort of related to this. It's induced, basically. Okay, so let's talk about natural genetic variation. It's easy to find. Go to your supermarket, look hard enough, you'll find stripes on purple bananas, or this sort of apple, or this happens to be a mutation. I used to work on tobacco when I was a graduate student. Here's a mutation I found. So mutations happen, and they happen all the time, and it's easy to see them if you're looking for them. Or you can see all the genetic variation if you go to sort of the home of some of the food that we eat, like potatoes in Peru, for example. A lot of variation. These are the tomatoes I grew in 2014. I have to say I grew almost the same varieties this year because they were so good. And these are all just various, variants on, I can't really, there's some tiny guys down there and some, big ones, and ones that don't even turn color when they ripen, and so on and so forth. So some of the genetic variation with tomatoes. As an aside, tomatoes actually have very little genetic diversity. Almost all the genetic diversity in tomatoes is for things related to fruit development. Otherwise, they're pretty genetically uniform. That's not surprising, right? So they went through a genetic bottleneck, and of course humans, however, select for, enrich, and save genetic variation for the traits that we're interested in, and we're interested in the things we eat, namely fruit. Okay. How many of you eaten string beans? How many of you have removed a string from a string bean recently? Then you're rare. because almost every bean that you buy is stringless. In 1894, Burpee discovered the stringless mutation, which removes the string, which is the fiber that occurs along the bean, which is why the string bean got its name, right? In the old days, you break it off and pull off that string. We don't do that anymore because it doesn't exist. Stringless mutation. Okay. Do you know why sweet corn is sweet? Why do you think sweet corn is sweet?
Because it doesn't metabolize the sugar in it.
It doesn't transform the sugar into starch. Exactly. Right? So field corn, the endosperm, which is where the starch is stored, of course, is the starch. And if you mutate enzymes in the biosynthetic pathway of starch, and starch, of course, is just glucose molecules stuck together, end on end, like glycogen, right? You mutate the enzymes that do the addition of glucose one to another, you end up with glucose, which is sweet, right? And here's one of the mutations this is the sugary 2 mutation. So if you buy your traditional sweet corn, it will be mutant for this gene, sugary 2. If you buy the super sweet corn, which is grown in the winter in Florida, it's shrunken 2, which is a different mutation, which really blocks starch biosynthesis, and that's really super sweet. I mean, it's actually not very tasty because it's just too sweet. So I won't go into the polygenic traits, but things like oil production and yield and other things like that, those are polygenic traits where lots of genes are involved, but we use single gene mutations quite commonly in plant breeding. This is the other important genetic discovery that is the basis for basis for the great increase in the productivity of American agriculture, which occurred corn agriculture, which occurred in the 30s. The effect of inbreeding was first identified in corn. Okay, so geneticists at the University of Connecticut and Harvard discovered if you self-pollinate corn plants, they got weaker and weaker and weaker by inbreeding. But they discovered that if you take two genetically homozygous or homogeneous plants, two inbred lines, and cross them and plant out that hybrid seed, the hybrid seed would give you this, hybrid vigor, heterosis, right? And so all the corn that you see, wherever you see it growing, wherever it's growing, is hybrid. It's uniform, right? It's all the same height because you have two uniform things, two inbreds, and you cross them. You get something that's heterozygous for a lot of things, but it's also genetically uniform and much more vigorous. So the discovery of hybrid corn, or the invention, if you want to call it that, along with other kinds of changes in the way we produce food after World War II, including direct mechanization, that is more tractors, new varieties of crops like the hybrid corn, fertilizers, herbicides, and pesticides, irrigation, and what have you, all led to a dramatic increase in productivity of U.S. agriculture. This is where we were up until about 1930, and after World War II, it took off. Okay? So the industrialized, high-yield agriculture that we take for granted in the United States is really a relatively recent creation. Okay. However, this change was not happening in the rest of the world. In 1960, it was recognized that the world was in the middle of a food crisis. There were lots of hungry people around the world. And the then head of the FAO, the Food and Agriculture Organization, got the UN to say, we need to do something about this. We should alert the world of this. And one of the ways they did this, I like to say this is the only time that every country in the world did the same thing. The UN got 140 countries to publish a stamp. And I have every single one of them, actually. And they all said, freedom from hunger. The United States also published a stamp. But you'll notice that the freedom from hunger is a small print, and what's up here is food for peace. Because we wanted to advertise the fact that we had a political program going on for 10 years where we tried to give food away to the world because we were worried about the communists. This is after World War II. And so we wanted to advertise that the U.S. had this, food program because we were afraid that if people were hungry, it would destabilize. There'd be overpopulation, resources, hunger, political instability, communist insurrection, so on and so forth. Okay, so when it comes to food, it comes back to politics. So this concern about food production, and the U.S. government, we couldn't sort of get it through Congress, but the Ford and Rockefeller foundations recognized this problem, and they created two institutes, one in Mexico called CIMMYT and one in the Philippines called the International Rice Research Institute. And these two guys right here, Borlaug predominantly, but then Henry Beecher, were basically, and it's true, the fathers of the Green Revolution. And what did they do? Well, they took advantage of some gifts that we got. One gift was this variety of rice from China, Di Jiao Wugzhen, which happened to be a dwarf variety of this rice. And it was discovered that this dwarfing was caused by a single recessive mutation. This is many years later. Okay? A single recessive mutation in the gene for the production of the hormone gibberellin, which is the hormone that causes stem elongation in plants. When this gene was cloned, it was said to be the most valuable gene in the world, and it's true. Probably saved more lives than anything else that humans have ever done, okay, because of it's saved off starvation. Here's another gift. This was a gift from Japan, although they didn't really give it to us. After World War II, when MacArthur occupied Japan, one of his generals collected 10 varieties of rice from Japan, brought them back to University of Washington in Pullman. You go to Pullman, Washington, you'll see a little plaque there honoring Norin 10, because Norin 10 variety from Japan had two dominant mutations in the gibberellic acid signaling pathway. Okay, so the first mutation in rice was a biosynthesis of a hormone. These mutations were the response pathway to that hormone that also caused dwarfing of the. And these were important for the breeding program because they were genetically simple. They weren't polygenic traits. These behaved like Mendelian traits, which meant that you could quickly introduce them into high-yielding varieties in a matter of a few years. It's just amazing. It wouldn't have happened if they weren't genetically simple. So why is having a short plant good? Because if your plants aren't short and you have a hurricane, or they call typhoons or rainstorm, and your plants are tall, the wind blows them over, and you have what's called lodging, and now your seed heads are in the mud and they rot. Okay? So if you have short plants like this, then that doesn't happen. And what that means is that you can then add fertilizer to your plants, and that fertilizer goes not into making a tall plant that falls over, but into making more seeds. Okay? Then this is, well, I was in the Philippines when this happened, 1966. This is when IR8, which is called Miracle Rice, which was the first dwarf variety of rice, was introduced. Here's the original variety. This is an Indonesian variety, Pita. And this is the variety into which that gene from the Chinese variety was introduced. And you can see nitrogen applied per hectare, the yield of PETA dropped because it got tall and it fell over, but IR8 increased. Marcos, who was president of the Philippines at the time, recognized this, publicized it, distributed the rice, and got elected the next year. This is what happens in terms of India and Pakistan. Okay? These are what yields were in the 1950s. This is Mexico, and here's India. Because Borlaug originally developed his varieties of dwarf wheat and also disease-resistant wheat in Mexico, but because he had unintentionally selected, by the way he bred them, for photoperiodic insensitivity, these varieties could be grown in many places in the world, including India and Pakistan. And here's what happened in terms of yields after that period. So it's important to preserve natural genetic diversity. In the United States, we preserve natural genetic diversity in this place right here in Fort Collins. That's the National Center for Genetic Resources Preservation. We have minus 80 freezers galore there. About 500,000 samples, seed samples, are preserved in Fort Collins. They also have vegetatively propagated plants. In 2008, Norway realized that it was important to preserve natural genetic variation, so they had an island they weren't using, Svalbard, about 700 miles from the North Pole. And so they asked places like our seeds, send us your seeds, and we'll store them in this permafrost. So they built a storage chamber back in this mountain. Here it is. And that's where the seeds are. And then, if the world ever has a nuclear holocaust or something, you might think it's far-fetched that you would need this. 2015, Syrian civil war, first withdrawal from the bank. Right? Syria, they lost all their crops from the civil war. And then this last summer, I don't know if you read it in the news, but due to global warming, things started to melt. And there was a lot of concern because they had built this hallway, not like this, but like this, and there was concern that the water would run back into the vault. Okay. So global warming is problematic for lots of reasons. Okay. So if you don't have any natural variants, what do you do? Well, of course, you can create them, right? So here's an aerial view, Google Earth, picture of a prefecture in Japan, Hirachiyomiya, I used to think this was a golf course until I counted how many of these, and there's more than 18, so it's not a golf course. But what I want to draw your attention to is not that, but that. That's a gamma ray facility. So sitting in the middle of that field right there is a source of ionizing radiation, gamma rays. Okay? And then you can plant whatever you want to irradiate at some distance from that and then expose it chronically to irradiation. Okay? So that's how you can do mutagenesis of large plants. If you have a small plant, like I used to do my mutageneses in radiation therapy departments. When I was a graduate school, I'd drive across New Haven to the hospital, and I'd take my plants into the, it was really interesting. You could go up there and you say, I need to irradiate my corn plant, or my tobacco plant. Where do I? It says, down the hall, it's Siemens Stabila plant, let me show you how to use it. Boom, 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 bang, And I did this at the University of Chicago too, and when I was here, I forget which department, immunology or something, we used to have a gamma source someplace in Richard's building where I would irradiate my seeds or whatever. You can't do that anymore. The machines are way too sophisticated. But anyway, so what are some of the mutations you could create? Well, if you buy a Mr. Goodbar, you're benefiting from this. So Hershey's recognized it would be good to have long shelf life for things that contain peanuts and things where oil can go rancid. So linoleic acid is the oil that gets oxidized most frequently. So, and that's produced by FAD2, and it was a duplicate gene situation. So they had a mutation in one of the FAD2 genes, and they mutagenized to get a mutation in the second gene. So now you have low linoleic acid peanuts, so that now you can keep them in your vending machines longer. Pink grapefruit. The pink grapefruit that we eat is an X-ray induced derivative to increase the pinkness of Rio Star. Okay, so that's an X-ray induced mutation. Okay, so you can create various mutations. In fact, Asians have, in Asia and China in particular, they've been doing this much more than we have in the United States, but that's certainly something that's been done. So the last method by which we can create genetic variation, of course, is using transgenes, so-called GMO technology. So the food that we eat in this country has had most of the certain crops, as I'll show in just a second, have had genes introduced to them by one of two methods. The original method, well, there are two methods. One is by using basically a gun to shoot DNA into cells. But the method that's more efficient takes advantage of this bacterial disease called Agrobacterium tumefaciens. It was famous actually as a plant example of cancer, because these are basically tumors. Okay? They're tumors that have unregulated cell growth, and they are tumors because basically bacteria, this bacterium right here, Agrobacterium tumefaciens, has introduced into the plant cell, by essentially mating with it, genes for the production of plant hormones that then cause cells to divide in an unregulated fashion. And along with that, it has introduced genes for the production of amino acid, which serves as a nitrogen source of the bacterium. But you can take this DNA that's transferred, the so-called tDNA, and get rid of those hormone-producing genes and get rid of the genes for the biosynthesis of the amino acid and put into whatever you want, green fluorescent protein, resistance to Roundup, whatever, and get the bacterium then to transfer the gene for you. This occurs so efficiently that the way we do, we make in my lab daily, if we want to, thousands of transgenic plants. In the plant that I work with, Arabidopsis, you just dip the plant in the agrobacterium. Take the plants when it's flowering, put the flowers in agrobacterium, take them out, wait a couple of weeks, sow the seeds on a selectable marker, boom. Whatever your marker is, resistance to herbicides, or I use fluorescent markers, there you have it. Hundreds, thousands of seeds, transgenic. No muss, no fuss. So easy to do. One of the very first transgenic plants was produced by Dr. Andy Binns, who used to be our vice provost and was the guy that sort of hired me in the biology department. And they put the gene from Bacillus thuringiensis. So Bacillus thuringiensis is a bacterium used in organic agriculture. It makes a protein called the Cry1A protein that basically punches a hole in the gut of lepidopteran insects like boll weevil or hornworm. And if you take that gene from Bacillus thuringiensis and introduce it into a plant like a tomato plant, which is what Andy has done here, it will be resistant to hornworm, whereas this is the non-transgenic plant. The other kind of transgene that's been put in is a resistance to Roundup. So here are some corn plants that have that Roundup resistance genes, and here are all the weeds around them that have been killed by the application of Roundup. So this is where we are, just to give you some sense. Here are the three major transgenic crops that are grown in the United States: corn, soybeans, and cotton. In the U.S. now, they were first introduced in 1996, and now as of 2016, we're about 95% of these crops. So if you buy anything in the United States that has soybeans in it, corn in it, or you're wearing cotton clothes, unless it says on the package that it was organic, it's GM. Guaranteed. Guaranteed. Now, the GM label, the organic label, part of the negotiations that went on in terms of how do you get a U.S. government-approved organic label on it was for the food not to be GM. Although in actuality, the presence of, for example, BT means that you don't have to put a chemical on the plant. But anyway, that was part of the negotiation. So the presence of a transgene is considered to make something non-organic in the United States. And it's not just one gene. This is a seed list I picked up at the Pennsylvania Farm Show a couple of years ago. And these are the seeds that are others. These are what are called hybrid trait classifications. They have trait brand name, trait, none. I love the idea that you can have corn with no traits. No transgenic traits. Okay. Here's a good one. This is Genuity Smart Stacks RIB. It has 7 transgenes in it. Roundup resistance, so it's resistant to Roundup, resistance to the herbicide Liberty, 3 genes for corn borer resistance, and 2 genes for resistance to rootworm. Okay, 7 transgenes.
What does it taste like?
Ask a cow. This is field corn, right? This is not sweet corn. Of course, as you all know, you can make transgenic animals as well. You can put whatever you want in them, green fluorescent protein. I bought some glowfish. These are the first transgenic animals available for purchase. And my Mary Mullins and Mika Granado, who are our neighbors, are there in the genetics, should be cell and development department, or whatever we're calling the department these days. I wanted to have some transgenic zebrafish to sort of one-up them. They have transgenic zebra in their labs. I want them in my home, so I bought some of these glowfish. But now you can buy transgenic salmon. This is the first transgenic animal, food animal, that is or will be commercially. It was approved by the FDA, I think, last year. This salmon right here contains a growth hormone gene from the Chinook salmon under the regulation of this promoter from the antifreeze protein gene from the ocean pout. So you basically a promoter that has high level expression of a growth hormone gene gives you a bigger salmon faster. So you get the market weight. These animals are actually, the genetics of these is really interesting. In order to get approval, they basically have a system where they create only females. And they're triploid. They create triploid female fish, and they raise them inland so that they can get around the idea, get around the issue with genetic, pollution or whatever of the transgenes getting out. They do that by pressure treating the eggs to basically diplodize them and what have you. And of course, the latest technology on the block is CRISPR-Cas9. And I'm sure everybody here knows about it or has heard about it or uses it. But of course, it's got lots of uses in agriculture as well. You can go onto the DuPont Pioneer website, as I did this morning, and they will direct you to, they have a whole website here that if you're interested in the technology. What is CRISPR-Cas? Why is it needed? How does it work in plants? So, will they be safe? So on and so forth. So the very first commercially available CRISPR-induced mutation will be a mutation in the WAXY gene, which is the gene in starch biosynthesis that basically produces a wax that's useful as a glue or like you're coating on your glossy paper from WAXY. So they're basically using this technology to create mutations in something you don't eat but can use commercially. Interesting, I was in Europe a couple of months ago just talking about this at this conference, and half the conference was about site-directed mutagenesis and use of CRISPR-Cas9. And it turns out in Europe, one of the issues is how big the mutation is. Apparently, if you have a small mutation, it can get around regulation easier than if it's a large mutation. And I like to give people the following example. The spontaneous mutation in the semi-dwarfing gene that created dwarf rice that I showed you, the green revolution, that mutation was a 380 base pair deletion. You can create an allele of that gene, and they have been created, using EMS mutagenesis by humans with a 1 base pair mutation that has, of course, exactly the same effect. So I ask you, which is the more moral gene? the big mutation that Mother Nature created or the small mutation that we created? Stupid question, isn't it? There's no logic to that question, right? Okay, so I'm going to end in the last 2 minutes here on some of the things that have not been approved. Here's one, golden rice. Okay, so this was created by some scientists in Belgium who wanted to create basically rice that had high levels of vitamin A. So they put the genes for carotenoid biosynthesis in the rice to make it yellow. Now, one of the objections to this is, we don't know what's going to happen if you eat, plants that have never had beta-carotene before, and maybe the intermediates will be bad for you. And I say, this is kind of interesting. We eat yellow corn all the time. We prefer yellow corn to white corn. We eat carrots all the time. They're good for your eye. Interestingly, some plants, high levels of beta-carotene are considered really good for you. Why is it in rice all of a sudden it's suspect? Right? Because it's never existed before. By the way, the mutation that caused carrots to be yellow is pretty recent, about 300 years old. Wild carrots are white. They're not yellow or orange. They have very low levels of beta-carotene normally. Okay. In Uganda, bananas are the main food source. They're like starch source. They're like potatoes to Ireland or rice in Asia, right? But bananas are susceptible to all sorts of diseases. If you buy bananas that are not organic, they've been sprayed with all sorts of things to keep them mostly protected from this fungal disease right here. And there's a bacterial disease that does this. This is the disease that wiped out the Gros Michel, nearly wiped out the banana industry in Central America, the banana disease. So there are transgenic solutions to this problem, and I think to this problem, but they're not available yet because of the issues relating to release of transgenic, particularly in some places like Uganda where regulatory mechanisms are not well developed. See, this is for that Xanthomonas resistant banana. So it still is not grown. Here's one, the EnviroPig. This was developed in Canada. The idea was to make so pigs release a lot of phosphate in their feces and urine. So the idea was to make an environmentally friendly pig by basically engineering it to metabolize phosphate more efficiently. Okay? And they did this by using a transgene, but of course this was resisted, no GM animals, and so it's not released. Even though the only reason for creating this was to improve the environment. So we have as humans this interesting dilemma. We can eat whatever we want. So we have, this is, by the way, this is not Michael Pollan's word. This is Paul Rosen in our psychology department, right? This is his line, omnivore's dilemma. You can go to the supermarket and you have choices. You can buy cornstarch that's 100% pure and GM. Or is non-genetically modified. It's up to us. That's it. Thanks.
Tell me what organic means.
Well, I mean, the United States. Yeah.
I thought it was just meant, had carbon in it.
So, right. So, of course, organic in the U.S., this is why it gets very tricky, because organic in the U.S., if it's labeled organic by the USDA, has a very specific meaning. Long list of regulations having to do with you can't treat plants with certain chemicals or herbicides and so on and so forth. It can't be GM. They can't be GM by this, by the method of putting a transgene.
Genetically modified.
By the addition of a transgene, So that the USDA, and I say to their credit, if you go to their website, they advocate the use of two terms, genetically modified and genetically engineered. The genetically modified term should be applied to essentially everything we eat. The genetically engineered term they would like to restrict to the use of plants and animals that have been engineered by the addition or modification via the technologies that we talked about. Right, so organic has in the United States a very specific meaning. Now, lots of farmers, and it also has regulations about what you can and can't do. So many farmers who are fully in line with growing organic food, don't want to be certified organic because it's too expensive. So if you go to a farmer's market and you ask some of the farmers, you say, is your food organic? Many of them will say, well, actually not technically organic in the sense that we're not certified, but we don't use any chemicals on our whatever, so on and so forth, right?
So the question should be, do you use pesticides?
You use pesticides, you use herbicides, and I can tell you the difference. We bought sweet corn. We have one's an Amish farm, one's a Mennonite farm. The Amish farm says, we're not really organic. We bought that sweet corn from the Amish non-organic farm, and they honestly say, well, we spray our ears, sweet corn ears, to keep the earworms away, are beautiful, never an earworm in those ears. The corn from the Mennonite, farm, every single ear had an earworm in it.
On my way down to Schuylkill today, I was listening to a program on NPR. And it was all about the use of antibiotics and the fact that 4 times as much antibiotic is used in agriculture
Yes.
than is in
On animals.
treatment.
Yes.
And I wondered, you haven't mentioned the word antibiotic. Is this relevant to what you've been talking about, or does the word ignoring pesticide? Would you care to address
Sure.
antibiotics in this context?
Sure. I mean, right. I left that out entirely, right? So antibiotics are heavily used in the animal industry, particularly in situations like the poultry industry and the beef industry, where they grow animals at high density in confined, so-called confined feed operations. So animals are treated with antibiotics like chickens, even before they're sick, as a sort of a prophylactic, and it increases yields by about 10 to 15%. Okay, and the idea is, if you grow 20,000 or 30,000 animals in one confined operation, the risk for disease is high. Okay, so it's absolutely true that the use of antibiotics in agriculture is the main use of antibiotics, and of course there's all sorts of problems with that relating to the development of antibiotic-resistant bacteria, so on and so forth. That's a consequence of the way industrial agriculture is. With respect to plants, interestingly, the use of antibiotics in plants really occurs at the stage of selecting for the transgenic plant. Because the selectable markers that are used to identify those, if you're making a transgenic corn plant that's Roundup resistant, Roundup, it's not so important because Roundup itself is your selectable marker. But if you're making a plant that you've introduced some trait in, you've got to identify the plants that have taken up your gene. And oftentimes they use an antibiotic resistance gene like kanamycin or hygromycin as the selectable marker. The transgene contains that. So antibiotics are used. So many of the, I don't know what they're using now in industry, but many of the transgenic food that we eat actually contains an antibiotic resistance gene in it because that went along with the transgene that you were putting into the plant. Okay, yes?
Have there been any documented human diseases that have resulted from GMO?
Right. So the National Academy of Sciences came out with, I think, their 5th report on this about a year or 2 ago, and specifically addressed this question. And the way these studies were done, of course, these are epidemiological studies. So the way they're done is you have to compare the United States, where we've been eating GM food since 1996, with Canada or some other country of similar, and there is no evidence whatsoever of any human disease or, that's a consequence of GM food. That, if there's one thing that can be said with some certainty, that can be said with some certainty. There are other issues with GM food. There's ecological issues. We can go on, and I'm not trying to say there are not problems. There are many problems. But I don't think human health is one of them. Yeah.
How effective are these organizations fighting against the production of GMO? I know there are companies that put their own imprimatur on food that's not GMO.
Mm-hmm.
Are they having an impact, or is it just a fringe group?
I think they're having a huge impact, certainly in Europe they have a huge impact, right? Their GM food is not approved in Europe, and it's really, to the extent that plant scientists, can't even use it for research, right? There's very, very negative. It's ironic that the very first GM plant was introduced in Britain. It was a tomato, actually. It's not grown there anymore. But so there's very negative feelings about GM, and the reasons for that are many. And I think, frankly, one of the reasons was that Monsanto overplayed its hand early on, and so there's an attitude about GM food that's mixed in with sort of an anti-American or anti-technology attitude, I think, that's built in there. But I would say that they're having a very large effect in many parts of the world, and even in the United States. I think people who, a couple of years ago weren't worried about this sort of thing are now are concerned about GM food. Americans are don't get me wrong. I hope you haven't. I'm sure I sounded like I'm a proponent of GM food. I'm not a proponent of GM or non-GM. I'm simply trying to say, well, this is what it's about, and this is how it's created, and you can decide whether you want it or not. And again, people's views about this will completely depend on people eat food for lots of different reasons. And GM, the studies that have been done in the United States on the impact of GM corn and soybeans on agriculture are that it hasn't increased the profits of farmers very much. It's decreased the use of chemicals a little bit, pesticides. Of course, Roundup use has gone up, but we've applied lots of herbicides anyway. So the net effect of GM on U.S. agriculture has not been tremendous. If tomorrow the U.S. government says no more GM corn or soybeans, we wouldn't suffer that much. Okay? It's mostly a matter of convenience for the farmer. Yeah.
Sounds like if you use a GMO cow, doesn't use that.
Right. It's interesting. If you talk to the folks at the New Bolton Center, so when I teach my food class, I don't know if you know this, but the University of Pennsylvania owns a dairy, Marshack Dairy at New Bolton Center. So I take my food class down to, I always tell them, you don't think Penn's an ag school, believe it or not. And so they have a 180 cow dairy there. And it's interesting to talk to the folks there because one of the arguments is, the argument for using, what's the main argument for using RBGH or RBST, to basically hormones in cows to increase their productivity? If you talk to them, the main argument for using that is decreased methane, environmental. And if you made a cow more productive, you would need fewer cows, right? And so, in fact, it's true that the size of the Holstein herd, the cow herd in the United States, I forget what the number is, it's dropped dramatically, since the 1950s because our cows are now way more productive. So from an environmental perspective, you could say, improving cow productivity has been environmentally beneficial because the amount of methane in the atmosphere is reduced. But you're right, if you made a cow that somehow, did not have methanogenic bacteria in its gut, or somehow you could modify it, that would be a huge impact. To answer your point, yes, it would be good.
In lieu of the fact that there's no evidence that GMO modified products cause any disease, why is there such an outcry against it by people who don't think for a second about ingesting all those foods filled with chemicals?
Right. I think it has to do, as I always say, with 2 million years of primate evolution. Actually, 200,000 years of human evolution. And what I mean by that is that animals in general and human animals in particular are very nervous about what you put in your mouth because it might kill you. So we have been selected over hundreds of thousands of years of evolution to be very cautious about what we eat. Okay, so those genes are in us. Psychologists have a word for this, So neophobia. Yeah. So I think that our anxiety about this is an anxiety about things we don't understand, particularly things we eat. We don't have such anxiety about using computers or other new technology or something that we don't put in our bodies. But things we put in our bodies, we have nervous system about, except when it comes to medicine. People have no problem, injecting themselves with insulin that was produced in a bacterium, right? Or any other drug that was produced in a genetically modified organism. Why? Because if you're sick, you want to get better. It doesn't matter where the drug comes from, right? But if you're healthy, you don't want to get sick, so therefore you get worried about doing things that you do not fundamentally understand or don't, So I think it's interesting, the gluten-free craze is one that I think is completely nuts. Right? That's an interesting problem, right? Because gluten is associated with disease in some individuals, for sure. But of course, we've been eating gluten for 10,000 years, right? And most of us do it quite happily. Yeah.
Are there any examples, or there are examples of where chemicals can cause mutations?
Yes.
And as a previous examiner for the National Academy of Sciences,
Right.
I had to inspect a lab in South Carolina
Yeah.
where they save all samples of every form of sea life,
Yeah.
eggs, fish, so on. And one of their concerns is that there is a steady growth in the content in fish, particularly and seals in the estuaries, of flame retardant.
Yeah. Right.
lost through all sorts of ways.
Right.
In estuaries,
Yep.
dumps, etc. Now, a thing like flame retardant is a pretty potent chemical.
Yeah.
And I just wondered whether there are any indications of where chemicals which have been introduced into our community by humans,
Yeah.
which were not naturally formed,
Right.
are also contributing towards, or could, mutate.
I don't know about mutations, but I have no doubt that the chemicals that are in our environment are causing, they could be causing autism for all we know, are causing our health hazards. I mean, you can tell, as I'm sure you know, what drugs people in cities are using by looking at the water supply. We can tell what the fraction of people in Philadelphia using cocaine is or whatever, but just analyzing our water supply, right? I mean, and I'm sure, we are so surrounded by chemicals in everything we do that it's undoubtedly affected many aspects of our biology that we don't understand. We just don't know what they do because we don't have a controlled population, right? If we had so I have no doubt. Now, with respect to mutations, probably everything causes mutations at some dose. So there's the attitude that basically some people believe that when you go to the supermarket, you should have, in addition to the price per pound or whatever, you should have probability that this causes a mutation, and then make your decision based on what level of risk you're willing to take, right? So the Ames test. I mean, Bruce Ames at Berkeley, he was the one to, he had developed this bacterial test for testing mutagenicity, where you take whatever you're concerned about and put it on these bacteria that were hypersensitive to mutations, and then you could get some estimate of its mutagenicity. And this is his view. His view is everything you eat, not just synthetic things, but plants or whatever you eat. I mean, to be honest with you, natural chemicals, for example, my favorite example is celery. Celery seems completely and totally innocuous. But celery has high levels of psoralen, which is a UV-sensitive molecule that can cause mutations. And when they initially selected for varieties of celery that were resistant to insects, they unintentionally selected for varieties that had high levels of psoralen in them. They were mutagenic, right? So they realized that and they backed away from that. But plants have been producing toxic chemicals to ward off insects, for a long time. So yes, so everything in our environment is potentially bad for us. Okay. All right, you probably want to get out of here.